Uh, Welcome, everybody. Uh, Welcome to New Approaches to 3D Vision. It's really fantastic to see such enthusiasm for this topic. It's wonderful. My name is Pamela Smith, and I'm the director of the Center for Science and Society. And this speaker series is part of the Presidential Scholars in Society and Neuroscience program. Uh, and it brings together interdisciplinary perspectives on mind, brain, and behavior research. And we're absolutely thrilled this time to be joining forces with two Columbia partners, the Italian Academy for Advanced Studies in America, and our director, the director is here, David Friedberg, as well as the Zuckerman Institute. And we're just thrilled to partner on this exciting event. If you're new to our seminars in society and neuroscience, I want to take just a minute to share just a little bit about our Presidential Scholars Program and the Center for Science and Society. The Presidential Scholars in Society and Neuroscience is a pioneering initiative that brings together early career postdoctoral scholars with renowned faculty experts across neuroscience and the humanities, arts, and social sciences. The program fosters cross-disciplinary research to enhance our understanding of mind, brain, and behavior, and it provides valuable support to postdocs and their mentors. In addition to offering seed grants for research, the program also hosts seminars like this one, conferences and events, promoting interdisciplinary collaboration and the exchange of innovative ideas. The Center for Science and Society, the home of the program, is a dynamic hub that connects researchers, scholars, and practitioners and aims to bridge the gap between traditional disciplines as well as enhance public understanding of science. Well, we are having a semester finally after pandemic years packed with exciting events. Our next event is a conference on the philosophy of deep learning brought to you by the PSSN program and New York University's Center for Mind, Brain, and Consciousness. And this will be your chance to really dive deep into the latest AI research challenges at the same time as exploring all the philosophical dimensions as well. You can register by scanning the QR code on this slide or by heading to our website. We also have coming up the Curiosity, Creativity, and Complexity Conference. It is now calling for abstract submissions for its poster sessions, so please submit your abstracts by March 1st and take the opportunity to showcase your work in front of a very diverse audience. And for those traveling from outside New York, a limited number of travel awards are available to help offset travel expenses. And we especially want students and postdocs from historically represented groups and research institutions to apply. So please learn more about the application process on the website, where you can also stay in the loop by checking out the Presidential Scholars website here for upcoming events and signing up for the monthly newsletter. We also have recordings of all our seminars like this one over the years. So there's a huge repository of extremely valuable material there. And please follow us on social media as long as it lasts and check out the updates. Little housekeeping here. For the online attendees, please use the Q&A button at the bottom of the screen. As you probably know, you can also upvote questions from others to make sure that they get asked. Our moderator, I guess me, actually will be sharing your questions. For those of you who are attending in person, well, you can just raise your hand, but wait for the microphone. So that everybody online can also hear. And we do have hundreds of people online. So we look forward to your questions. I just want to say that the success of the PSSN program is due to the really unwavering support of Columbia's leadership, the tireless efforts of our steering committee members, some of whom are here today, and the invaluable contributions of faculty from across the university who serve on the advisory committee mentor our scholars, and volunteer to review applications and to interview our applicants. All of your dedication has transformed this program into a really extraordinary example of interdisciplinary exchange and a true testament to the power of collaboration. I also want to take this opportunity to thank our wonderful, incredible administrative staff, Joseph Sulik and Melinda Miller. And now, without further ado, I'd like to introduce our moderator and presidential scholar for the coming 3 years, Paul Linton. Dr. Paul Linton is a neuroscientist and philosopher with expertise in 3D vision. He received his PhD in 2021 from the City University of London, where his research challenged our understanding of distance perception by showing the visual system is unable to triangulate distance using the two eyes. Dr. Linton was previously a lecturer in law at St. Hilda's College, Oxford, and a teaching fellow in philosophy at University College London. As a presidential scholar, he will develop his new two-stage theory of 3D vision using machine learning and fMRI in the hope of explaining how we experience the 3D world. Please help me welcome Dr. Linton.
Cool. Well, thanks so much, Pamela, and thanks everyone for coming today. And thanks also to the people watching on Zoom. So the first thing I want to do today is to say thank you to the sponsors of our event. So the Presidential Scholars in Society and Neuroscience, the Italian Academy, and also the Zuckerman Institute. And in particular, I want to say thanks to Pamela and Melinda, Joseph, and Caroline at Presidential Scholars, David, Barbara, Abigail, and Kathleen at the Italian Academy, and Daphna, Carol, and Mitali at the Zuckerman Institute. And really a special thank you to Joseph and Mitali, who've just been amazing in organizing this event. So the purpose of today's event is to launch the New Approaches to 3D Vision Royal Society volume, which was edited by myself and also by Fulvio, as well as Michael Morgan, Jenny Reed, Dhanraj Vishwanath, and Sarah Cream-Riga. And this built upon a previous Royal Society discussion meeting that we held that had speakers from academia, as well as from industry, so from DeepMind, Google Robotics, Microsoft Research, and Facebook Reality Labs. And all of the recordings are online for you to see as well. Good. So the purpose of the volume was to explore how artificial intelligence and computer vision, animal navigation, and human vision are all beginning to question how we sort of see and act in the 3D world. So what do I mean by this? Well, the traditional approach to 3D vision is that in order to act or navigate in the 3D world, you first have to build an accurate mental map, a sort of accurate 3D mental model of what the world's like, right? But recently what we've seen is increasingly computer vision, animal navigation, and human vision have all been questioning whether this is really necessary. So, for instance, would a distorted model work? Or perhaps no model at all? Good. So to explain this in the context of these three different disciplines, if we start with artificial intelligence and computer vision, for a long time, the focus in computer vision was primarily on LiDAR and SLAM, right? These are methods which essentially produce high-resolution 3D maps of the environment. But more recently, we've seen a resurgence in attempts to build robots that navigate without building an explicit or accurate model of the world. Similarly, if we want artificial intelligence to mimic human behavior, we need a more nuanced account of what artificial intelligence needs to learn about the environment and how it learns it. So Ida, in her talk, is going to explore how artificial intelligence can inform and be informed by neuroscience. Turning to animal navigation, the 2014 Nobel Prize was won for understanding the brain's sort of GPS system, right? And this relied on two big discoveries. So first, place cells were discovered in the rat brain that activate when the rat's in a particular location in the environment. Then, second, grid cells were discovered that cover the whole environment with these firing fields in blue that activate whenever the rat crosses them. So together, this combination of place cells and grid cells seemed to be that ideal metric model of the world that people thought was necessary for navigation. But what Kate's going to discuss is that actually these mental maps are much more distorted than we initially appreciated. And finally, human vision, right? So the recent revolution in virtual and augmented reality has turned attention back to human 3D vision. Here, again, the assumption is still that human vision aims to provide us with a sort of accurate or metric map of the environment. But the problem with this is that human vision often departs quite radically from the reality, right? It's subject to massive distortions, as Fulvio is going to discuss. And so rather than insisting that human vision is trying to, as it were, recover reality, Fulvio is going to argue that actually it's doing something else quite different. So that's where I want to start the discussion with Fulvio. So to introduce Fulvio. Fulvio is professor in the Department of Cognitive, Linguistic, and Psychological Sciences at Brown University. His research team investigates how the human visual system processes 3D visual information to enable successful interactions with the environment. His approach is to combine computational methods and behavioral studies to understand the visual features that establish the mapping between vision and action. And his research is funded by the National Science Foundation and the National Institute for Health. Welcome, Fulvio.
Okay, thanks, Paul and everybody for organizing this amazing event in this beautiful place here. And let's just start. So since at least 1,000 years ago, from the time of Ibn Haytham, also called Alhazen by Europeans, we've been trying to figure out how a 2D visual input like this video produces the perceptual experience of a three-dimensional world. And the traditional way that this problem is being approached is to ask the question, how does the brain infer the physical structure of projected objects? And in this talk, I will question this formulation of the problem. I will first briefly describe the Bayesian approach that tackles 3D vision as a problem of perceptual inference. I will then argue against these fundamental assumptions of the Bayesian approach, the fact that the brain derives the most probable physical structure that produces an image. I will then present an alternative theory that I've developed in the last few years, namely intrinsic constraint, which rejects this assumption, and I will outline the vector sum model, which implements this theory. And I will also briefly describe data, if we have time left, that supports the prediction of this model. So the traditional approach is based on the observation that there is an infinite number of physical structures that produce the same retinal projection. Fortunately, not all interpretations of the image are equally probable. If you know how images are formed, you can calculate how likely it is that a certain object projects a certain image. If you know what kind of 3D structures there are in the world, then you can have a prior information about the most probable structure in the world. And then through this, through the famous Bayesian theorem, you can then multiply likelihood and prior and associate probability to the 3D interpretations and choose the most probable. So this is certainly something that computer scientists know how to do in general. The question is, can the brain do it? Before we get there, let's see how this usually works. The image is decomposed into independent signals. For example, in this example, we have the disparity signal and the texture signal. Disparities are the differences between the images into two eyes, and these differences co-vary with the depth of points in the world. And texture information is given by how elements on the image are distorted and how these are related to the distal surfaces. So then independent modules calculate the probability of physical structures given the disparity Q and probability of physical structures given the texture Q. Then these estimates from these modules are combined in what is called a statistically optimal fashion to get overall probability of physical structures given the image. And this estimate will be more reliable than the single Q estimates. Okay, so these models estimate the physical structure, but what do we mean by physical structure? It really boils down to the relationships among measurements taken with a rigid ruler. These measurements will follow specific rules discovered by Euclid. So asking how the brain derives the physical structure of this cup is really postulating that within the brain or our mind, there is a geometrical structure obeying Euclidean geometry. And mental rulers have exactly the same readouts of the rulers they use to measure the world. The problem is that biological systems don't go around with rulers to measure things. In fact, biological systems are wet stuff that transduces, for example, light in electric energy or transduces soft mechanical pressure into electrical energy. And this is how the system is being bootstrapped to be successful in performing actions. So postulating then the brain can calculate how probable it is that a given physical structure can produce a certain texture pattern on your retinas or disparity field is a very big leap of faith. But this is exactly what most popular theories of 3D perception posit, that the system has somehow associated sensory information with the Platonic structure that we call physical structure. And the approach I propose do not require leaving the cave. As we can see here, we can avoid being hurt by the light of the Platonic world, and instead we can chill out in the cave cozily and observe the relationships among the signals in the cave. Without knowing anything about the Platonic world, we will notice that the pattern of distortions of texture elements and retina disparities are related to each other. And since these image signals are computed through completely independent processes, we will conclude that the correlation must have some common cause. In this example, I call this common cause depth. It is meaningful to the visual system because it also correlates with the hand grip that would successfully grasp the object. What I'm speculating here is that the visual system has evolved to discover independent image signals that are meaningfully correlated. And the latent variables that describe these correlations are what we call 3D properties. Of course, if the signals are related to depth, to each other, then they're also related to the physical depth from which they derive. So, but here's the catch. So, image signals do not only depend on 3D properties, but also on other factors. For example, if you look at a cup that is very far away, the differences in two eyes disappear. The disparities becomes very tiny. So the slope, what I call KD here, relating disparity and depth, vary with distance. And I will call this slope strength from now on. The blue area here indicates the range of disparity strengths. The strength of the texture depends on the material property. Bad material properties give you bad texture information. And here indicate the range of all possible texture strengths in green. Therefore, distance and material composition are undesired nuisance factors. And the goal there for the visual system should be to reduce as much as possible the influence of these nuisance factors. And this can be done by choosing a metric for this multidimensional space of signals that favors signals that vary less with the nuisance factors. And it can be mathematically be shown that the length of the vector that has these signals as components will maximally correlate with meaningful variables such as depth related to nuisance variables such as distance and material composition of objects. So now we are going full circle. We started with Euclid in Greece, back to the holy world of Bayes, back to the Hellenic world with Pythagoras. So let's now directly compare the assumptions and predictions of Bayesian models and the vector sum model. Bayesian models assume that depth modules are noisy, but give you on average accurate estimates of the physical ground truth. The vector sum model instead predicts generally biased estimates. Critically, it does not predict noisy estimates. Bayesian models combine a single Q estimate through a weighted average, where the weights are inversely proportional to the variance of the noise. This way they achieve statistical optimality. For the IC theory, our intrinsic constraint theory, signals are affected by a negligible amount of noise that does not depend on the particular cue. Therefore, it does not weigh signals. Instead, it produces depth estimates that increase in magnitude with the number of signals. Bayesian models predict that combined estimates are less noisy than single cue estimates. The vector sum model does not predict a reduction of the estimation noise as cues are added. Instead, combining cues reduces the influence of what I call nuisance variables. So let's start with the first two predictions of this model. A PhD candidate in my lab, Jovan Kemp, ran a study where participants judged the depth of a sinusoidal corrugation. You can see on the bottom, at the bottom. In one condition, the corrugation was specified by texture cue, as you can see on the left. And these are the results for this condition. On the x-axis, we have the simulated depth of the corrugation. On the y-axis, the perceived depth. On the left panel, I show the result for viewing distance of 40 centimeters. On the right panel, for 80 centimeters. For a lucky circumstance in this case, depth from texture is almost accurate at both distances. The dashed gray line indicates accuracy. In a second condition, only disparate information was available. Note how depth from disparity is actually overestimated at 40 centimeter distance. And there's a clear discrepancy with the texture information, right? And at 80 centimeters, a gain for a miracle, disparate information seems to be veridical. However, what are the predictions? So Bayesian models would say, well, if these are the perception from the single cues, then combined cues should be in between. However, in both cases, perceived depth is larger. At 80 centimeters, we have the paradoxical result, actually, the paradoxical result that the combined cue estimate is actually less accurate because it's more overestimated than the single cue estimates. And in gray, I show the prediction of the vector sum model without free parameters. You just plug in the numbers from the single cue conditions and you get those predictions. But the prediction of the vector sum model can be just seen with demonstrations. I guess everybody, I hope here, perceives this sphere that contains shading and texture information as deeper than these two that only contains shading or texture information. Now, let's turn to what I think is the fundamental difference between these two models. For Bayesian models, cues are combined to reduce the noise of depth estimates. For the vector sum model, cues are not combined to reduce the noise of depth estimates, simply because there is no such thing as noise of depth estimates. And critical now is to consider how the alleged noise of depth estimates is being measured in the past. This is done through what is called the just noticeable difference, the minimum amount of depth difference that a subject can discriminate between two stimuli with the 84% discrimination accuracy. Since it is assumed that the average estimates are unbiased, then the J&D becomes an estimate of the noise, of the standard deviation of the noise. This is the noise for texture information. This is the noise for a less reliable cue that is shading information. And this is the noise for a combined cue, which is better than the single module cues. And this is the most important tested predictions of Bayesian models. The variance of the combined cue estimate is smaller than the variance of the single cue estimate through this equation. And a large number of studies in the past have confirmed this prediction by using J&Ds as proxies for the standard deviation of the estimation noise. So this is a very important prediction. So now, let's start now to the vector sum model. The vector sum model predicts negligible noise of depth estimates. So where is the noise coming from? You have noise that is task related. When you have to memorize a stimulus and then remember the depth of the stimulus compared to another stimulus, then of course you're going to have noise associated with this. For instance, so there is a minimum, however, as I said before, there is a relationship between simulated depth and perceived depth given by the vector sum model that is given by the strength. So the J in D is the amount of physical depth difference between two stimulus that elicit a perceived depth difference that overcomes the task-related noise. So the J in D is inversely proportional to the Q strength. If you have a Q that is weaker, which I assume that the shading is, then the J in D will be larger. When Qs are combined, the signal is even stronger, as predicted by the vector sum. Therefore, the J in D is smaller. So I leave it to you as a homework to show that this J in D prediction is exactly identical to the one that was used to test Bayesian models. However, Jovan has adopted a new, very elegant method that can discriminate the two models. I let you guess which model wins and stay on the lookout for published papers. So the take-home message from this presentation is that perhaps we should look at the problem of 3D perception from a different perspective. I reject the assumption that through evolution or development, the visual systems be able to find a mapping from sensory information back to the physical structure of the world. Instead, I suggest that the visual system exploit meaningful relationships among signals to discover latent representations that only co-vary with distal properties. I argue that these latent representations do not provide a faithful copy of the physical world, but are sufficient for successful interaction with the environment. We have a lot of data that supports this theory. And finally, I want to stress that I do not reject Bayesian model per se. What I reject are the assumptions made in the past by Bayesian models for 3D perception. And I thank everybody for listening and the lab for helping me with all this. Thank you.
So now to move on from human vision to animal navigation, I'm going to introduce Kate Jeffery. So Kate Jeffery is a behavioral neuroscientist researching the neural representation of space using single neuron recording methods in rodents. After being based at UCL, University College London, for many years, she has recently moved to the University of Glasgow in Scotland to become head of school for psychology and neuroscience. Welcome, Kate.
So before I delve into the science, I just wanted to make a quick plug for Glasgow. So we are hiring postdocs, assistant professors. Come and join us. It's a really beautiful place. Really exciting to be there. So I'm not explicitly working on vision per se, but vision is very important for the system that I do study, which is navigation. And I'm going to be picking up the same theme that we have been thinking of how animals represent the problem of navigation, the problem of getting from one place to another. Thinking of it in terms of there being some kind of internal model of the world that the animal consults when it wants to get from A to B. And part of the reason for thinking about this is that in our own subjective experience, we feel as though we have an internal model of the world. So we feel like we can visualize, New York, whatever, and how to get from one place to another. But also psychology over the last 100 years or so has very much embraced this idea that was introduced in the 1940s by Edward Tolman. Based on studies of rats, he concluded that the way that rats were able to plan their way around to get food couldn't be explained by just supposing that the animals were looking at their sensory world and making simple associations between the world and the actions. The only way that he could explain the complexity of behavior was to postulate that the rat had some kind of internal map, and he called it a cognitive map. And that term has been very much debated over the years, but the idea that there is some kind of internal representation of space has been very, very powerful. And depending on who you talk to, there are various different formulations of this, and the various different formulations have caused a lot of very heated debate in the past few decades. So at one end of the kind of hardness scale, if you like, is the idea that this map is very much a metric map. So there are very precise distances and directions that are being, oh, sorry, I'm starting at the wrong end. So that there isn't a metric map. I'll put all of them up, how about that? So on the right-hand side, the idea that there's a metric map. So there's something very much like the kinds of maps that we use, which is that the representation has the same relationships between the elements in the map, like on the page or whatever, as in the real world. So that obviously the scale is different, but things like the angles are the same. And so you can make measurements on your map, and that will relate very directly to the things that you have to do in the real world. So that's the kind of metric map formulation of the cognitive map. And then at the other end, on the left, is the more kind of loose notion that really the brain is building up this internal representation that's based on just discovering relationships between things and discovering that there are certain regularities, like when I'm in place A, if I move, I very often get to place B, but not so often to place C, and so on. And so as you're kind of walking around, you learn the sort of relationships between places. And so you end up with this kind of fuzzy relational set of associations in your head, which acts kind of like a map. You can use it for navigating, but the precise distances and directions are not captured. And then there's something that kind of in the middle, which we call a topological map, which is halfway between. So as you're moving around, discovering places in the world, you kind of roughly know where they are in relation to each other and roughly know the angles, but not very precisely. So you couldn't make precise calculations of exactly which way to go, but you sort of know which way to go. And the adjacency relationships are all preserved. So that's the kind of soft version of the metric cognitive map, if you like. So neuroscience started to get in on the act in the 1970s with this very important discovery that was made by John O'Keefe at UCL, and he was deploying this new method for recording single neurons in the brains of rats that were moving around, which was a really new technique for the time. And he was recording from a brain area called the hippocampus, which was attracting attention at the time because it seems to be very important for memory, and people were very interested in memory. And so he was deploying this new technique, recording from the hippocampus. And what he noticed was that if you take note of the action potentials that are being emitted by one individual neuron, so these are the traces that you would see on an oscilloscope screen. For all the neuroscientists here will be very familiar with this. So each one of these is an action potential, a nerve impulse. And what he noticed was that if you take the action potentials and kind of plot where the rat was when the neuron emitted the spikes, as we call them, they would tend to congregate in one particular part of the environment. So as the rat's kind of wandering over this square platform, for example, every time it goes to this corner, the particular neuron of interest suddenly emits a little burst of action potentials. And so O'Keefe proposed, together with Linton, who was collaborating with him, who's a neuropsychologist, they proposed that what the hippocampus is doing is creating Tolman's cognitive map, so an internal representation of space. And they wrote this book called The Hippocampus as a Cognitive Map, which was very kind of enraging or enlightening, depending on the viewpoint that you had prior to that. It really galvanized the field a lot. So it seemed to be making a very convincing case for these place cells being the substrate for Tolman's cognitive map. So I'm privileged to be one of the few owners of a copy of this book, which has been out of print for a long time, and that's a picture of me getting it signed by John O'Keefe at the champagne reception for his Nobel Prize, which he got for the discovery of place cells. And I subsequently got Lynn Nadel's signature on that as well. So I have this treasured item in my possession. So it was a very, very kind of important idea. So the question then, if we think about these cognitive maps and whether they're metric, is the hippocampal cognitive map metric? And about sort of 15 or 20 years after O'Keefe discovered place cells, a new discovery was made that suggested that it might be metric, and this was the discovery of head direction cells. So this was made by Jim Ronk, and it was published in a little abstract in 1984, and then nothing happened for a few years. And then Jeff Taube came to his lab and was set to work to kind of characterize these cells. And so what these cells do is they fire whenever the rat faces in a particular direction. So we depict the activity of these cells by taking the 360 degrees of compass orientation and plotting the firing rate. So this one cell fired at a very high rate when the rat was facing to the southwest in that environment, and a very low rate in all the other directions. And different cells would have different directions where they would fire when the rat was active. So it looks like this part of the brain is kind of constructing some kind of compass for all of the directions. And there are sort of certain properties of this network that are very, very characteristic. And one of them is that the activity is very, very kind of stable from one moment to the next. So when the animal turns its head from, let's say, north to the right. And that cell that fired when the rat faces north would stop, and then the next one would start. And computational modelers propose that actually what's happening is that these neurons are all arrayed in a network. This is a conceptual idea. So let's say this is the neuron for north, and all the other kind of directions are here. And so as the animal is moving around, see, there's a video of a mouse in this case, moving around, activity is moving around this network in accordance with how the head is moving. So it's constantly tracking its movements. So it's producing a constantly updated signal for direction. So this is called an attractor network. It's got lots of interesting properties, which I won't get into. So that seems metric. So that the angles are being very precisely computed and conveyed to the place cells. Unfortunately, there isn't actually a direct connection from the head direction cells to the place cells, but that was a kind of a minor quibble. But we were pretty sure that the head direction cells were kind of the compass for the place cells. So that's kind of a metric for direction. But the question is, what about distance? Because if you're going to use a map that's metric, you need to be precisely recording the distances that you've walked as well as the directions in which you've walked. So about 15 years after the discovery of head direction cells, another discovery was made that really electrified the field still further. It was made in the lab of Edvard and Maj-Britt Moser by Torkel Hefting and Marianne Fern, who were PhD students. And they were recording from a region of cortex near the hippocampus. So this is the rat brain, this is the hippocampus here in blue. And they were recording from entorhinal cortex, which is the part of the cortical region of the brain that sends the densest inputs into hippocampus. And Menno Witter, who is an anatomist, very, very gifted anatomist, had said, look, if you want to understand the source of the spatial signal in the place cells, you should record from entorhinal cortex. So they did. And what they found was that entorhinal neurons, many of them have spatially localized firing that's quite similar to the place cells. So if you go back to the depiction method where a dot is a spike, an action potential, this is one of these cells, and it's producing a bunch of spikes when the rat goes here. So the rat's walking around a 2-meter diameter circular platform. So this black line is the path that it took. So every time it goes over here, the cell fires. But also it fires when it's over here, and it fires when it's over here, and it fires when it's over here, and here, and here. So this one cell has all of these places in the environment where it will fire. And the really striking thing about this pattern is that the distance between those blobs, if you like, is very, very regular. So in this case, it's about 30 centimeters. And also the directions of alignment of these rows of blobs are very, very consistent. So the rat's going backwards and forwards, but every time it comes to this region, that cell fires again. So the cell is tracking the distance and direction that the rat is moving. And that's a process that we call path integration. And it seemed to suggest that what these neurons are doing is calculating distances for the cognitive map. So that seemed to be very strong evidence that, and not just distances, but also directions. So very strong evidence that the cognitive map is metric. So when this discovery was first published, Neil Burgess and I kind of said, yay, we found the metric for the cognitive map, because up until that time it had been just a theoretical idea. And we started thinking about how to exploit this characteristic of grid cells, that they seem to be giving us a window into the metric structure of the cognitive map. And I got interested in the question of whether this map of space fills 3-dimensional space. So this is where the third dimension comes in. So we have what looks like a metric map. What happens in the third dimension? And we had various kind of possibilities for what might happen. It might be that the grid cells, as they were called, I should say also that the Mosers got the other half of the Nobel Prize in 2014, but they didn't write a book, so I haven't got any signatures for that. But place cells and grid cells together were regarded, and rightfully so, as transformative in our understanding of cognition. So the grid cells in 3 dimensions might not care about the vertical dimension. So it might be that their job is just to track the progression of the animal over the surface of the environment. Or it might be that that the same map that is produced by the grid cells, in other words, the same distance computations, take place at all the different vertical levels that an animal might traverse, or a human. So you go up to multiple floors of the building, your grid cells are still working, but in each level, they're still only working in the horizontal plane. So they're only tracking the distance that the animal moves in the horizontal plane. Or it might be that they can kind of take account the movements of the animal over the surface. So it's not so much the horizontal distance that they care about, but how far has the animal just walked, like how many footsteps or whatever. So if it's going over an undulating terrain, it's counting footsteps, and it will count more of them than if the animal is walking flat. And there is some evidence that might be possible. Or final possibility is that actually the grid cells have the capacity to track movement in all 3 dimensions. So really they can form this, it's kind of a Platonic ideal, getting back to Plato's kind of forms and so on. So they can form this kind of Platonic ideal of distances computed in any direction that the animal might go. So we started trying to look at this in my lab, and it's kind of complicated by the fact that rats don't swim or fly through 3 dimensions. But I had some very dedicated students and postdocs who developed an apparatus that animals would happily wander over, called the pegboard. So it's just a climbing wall with pegs sticking out of it. And we started off recording place cells on this apparatus, and then grid cells kind of got discovered after we'd started this experiment. So we added grid cells to the mix. And the question is, what did the grid cells and the place cells do? And we found something very surprising, which I hadn't expected at all, which is that if you look at the way that the spikes from one of these cells are distributed over the surface of this wall, so again, the black is the path of the rat, and it's a little bit broken up because of the pegs, but you can see that the rat was covering the surface pretty well. So the spikes, they're not forming blobs in that nice, neat kind of hexagonal close-packed array that they do on the flat. But there is a sort of a sense of periodicity if you look at the horizontal dimension, because there's some firing here, and then there's a gap, and then there's some firing here, and then there's another gap, and then there's some firing. So it sort of looks like in the horizontal dimension, the grid cells are still trying to produce blobs, but those blobs are continuous in the vertical dimension. So it's not forming a grid that permeates volumetric space, but they are doing something. So we thought maybe the Platonic ideal for grid cells is something like this. So if you look at the horizontal plane, I don't know how well you can see, the slide's a bit faded, but you can see the hexagonal pattern. But if you look at the vertical dimension, then you get these stripes. And the reason for the varying spacings between those stripes could be because that's what you would get if you randomly plonked a vertical wall in the middle, randomly in the middle of a field of grids. So then the question is, the distance tracking on the horizontal, was it really the horizontal or was it the surface? So we thought, well, let's just test this by making the surface not horizontal and seeing what happens. So we built what we call the gradient box, which is a box that has a flat half and a sloping half. And the question is, what would the grid cells do on the sloping half? And we thought that if the grid cells are just tracking horizontal distance traveled, then they'll do something slightly different on the slope. And I won't get into details of how for time reasons, but it'll look different. But what we found actually is that it doesn't look different, it looks the same. So for every one of these panels, each one of these is a cell. The left-hand side is the sloping side, and the right-hand side is the flat side, and it just looks the same. So the cells just seem to be tracking distance on the slope in the way they do in the horizontal. So these 2 things didn't seem to quite fit together. So we persevered, and we thought, okay, so it might be that the vertical on the wall actually has 2 different meanings. So the way that we had the rats is that although they were walking around kind of sidling around on this vertical surface, in fact, they were oriented horizontally. So what would happen if we actually made them orient vertically? So my student, Giulio Casali, spent a very, very long time training rats to walk on chicken wire on a vertical surface or a horizontal surface. And he found, the question was, would we see something that looks like blobs on the vertical surface now that the rat is walking over the vertical surface, or would we see stripes like we saw on the pegboard? And what he found was that it wasn't stripes, it was blobs. But what he also found was that the blobs are shown in the turquoise on each of these panels, so that's the wall. The red is the floor. You can see that it's blobby, but the scale of the blobs is huge, much larger than it is on the floor. So something weird is happening to the ability of the grid cells to track direction. So we've got all of these different patterns of what grid cells are doing. They seem to be doing things that are inconsistent with any one type of map. So depending on how we're getting the animal to move through the space and what kind of surface it's on and how it's oriented and so on, we get a completely different pattern. So there isn't one underlying structure for that representation that can explain that. It kind of looks like the map, if you like, is adapted to whatever surface, sorry, to whatever situation the animal is in. So that was all kind of surface travel, but then we thought, okay, what about volumetric travel? That's the final test. And that was a very, very technically difficult experiment to do, and it took us quite a lot of years to solve the technical challenges. But Ronnie Greaves finally managed. And so what he did was he got rats to explore through this lattice while recording the cells wirelessly, which was really a very, very complicated feat. And what he found was that place fields formed volumetric structures in that space. So the place cells seemed to be able to track the movements of the animals through the space. And grid cells formed blobs, but they didn't form regular blobs. They formed irregular blobs. So the cells, and they were stable irregular blobs. So the rat would walk through the space, and every time it returned to a place in that lattice, the cell would fire, in the same way. So it was a stable pattern. So the cell was able to track the movement of the animal through the space, but it wasn't positioning its firing in a stable way. So to sort of conclude, all of this raises questions to me about the assumption that we made at the beginning that the grid cells are forming the metric fabric of the cognitive map of space. It seems like animals and place cells can do very well without a regular grid, so long as the grid is sort of there in some form or another. So it can be regular-ish, or it can even be irregular and still work. It's seeming to be the case, and not everybody agrees with this. Some people do, that the important thing about the grid cells is not the regularity of the pattern, but something else, and the something else is waiting to be discovered. So I'll just sort of leave you with this picture, really, which is that the pattern that we get, the nature of the map, is tailored to the individual specifics of whatever environment the animal is in. So it seems to be constrained by the movements that the animal is able to make and the way that it's able to ambulate and so on. And it's really kind of constructed rather than retrieved from out there. And this is an idea that has become very relevant to psychology in the last few years. It's called enactivism, which basically means that your internal model of the world derives from your movements and your interaction with the world. So it's not just this pure thing that's constructed. You construct it by interacting with the world, and I think that's a really nice idea. So I'm going to just stop there, skip over to my acknowledgements, and thank you for your attention.
So finally, we're going to turn to AI with Ida Momennejad. So Ida is a principal researcher in reinforcement learning at Microsoft Research, New York. And this is the important point, she's also the co-host of the Learning Salon, an online weekly forum where every week they invite a new expert to discuss the connection between biological and artificial intelligence. So you've all got to sign up, okay? Anyway, please welcome Ida.
All right, with that little interlude, hi everyone. Excited to be back at Columbia. I've been here since the pandemic. It's been wonderful to see some colleagues again. I'm going to tell you a little bit about a rubric for human-like agents and neuro AI. For those of you who are less familiar with the field, I have added ample text. Hopefully the lack of aesthetics due to having too much text is going to be okay. First, I want to thank my students and postdocs who have collaborated on many of the projects, some of which you will see here. So thank you. If you ever see them somewhere, say hello. Just to give you a summary of what is ahead. People use terms like human-like AI, neuro AI, neurally inspired AI, brain-inspired AI. Sometimes they even interchangeably use these words with artificial general intelligence, but actually they don't mean the same thing. So some of the things that I'm going to do in this talk, in addition to showing you a little bit of how 3D vision actually informs some of the work that we do in Xbox, and 3D vision is not really my field. I mostly work on human spatial navigation and memory and build AI that does that. However, the agents that we build require 3D vision in order to do what they need to do. So this rubric is supposed to help to clarify some of the confusing crosstalk across field. For instance, in AI, the concept of transformers has enabled ChatGPT and all the buzz that you're hearing these days. And there is the notion of attention or self-attention in that model. And there is the notion of attention in human and animal research in neuroscience, and these two have very different meanings. It helps to identify what are the primary goals of individual projects in order to even understand what the terms mean sometimes. And so I'm hoping that this rubric can help some of the students out there or folks who have already been thinking about these things to think about the ancestry and the future of some of the research that they're doing with regards to the main goals. And finally, hopefully it'll allow us to come across what would be the future ahead. So what are the different priorities and goals that are going to happen? You will see all of these figures again. So I'm just going to go through it really quickly the first time around. I've identified a couple of things. The dimension of neural plausibility. You could build a model or an agent that's neurally plausible. For instance, if we wanted to build a navigation agent that's neurally plausible, it will have to be able to explain some of the neural data that Kate was showing earlier. And then if you want to explain something that is about human-like behavior, for instance, if you want to explain some of the behavior results that Fulvio was showing, your agent will need to be able to show the same kinds of accuracies, reaction times, and delays, and errors to what humans were performing. However, if you want to just solve some engineering problems, for instance, you just want to build a search engine that responds to people in words, like fuller sentences, then you don't really necessarily need to care too much about these dimensions. You can care a little bit, perhaps, just because you're interacting with humans, but not with the rigor that some of us who actually try to understand human behavior and brains do that. So let's take a look at the different dimensions that we considered. Agents could be built with the goal of being neurally plausible, with the goal of showing human-like behavior, or in case of some other colleagues, rodent-like behavior, cat-like behavior, bird-like behavior, bat-like behavior. Or they could be serving engineering goals. I'm not saying these three goals are completely separate from each other. They can, in fact, interact. A certain project can serve a number of these goals. However, it's very important to know they are not necessarily interlinked and they do not necessarily translate to one another. So what does that mean? It means that there can be generative, corroborative, or corrective interactions among them. Model with a particular goal of one dimension can help to corroborate a model in another dimension, or to correct one which had some kind of problems, for instance. However, success in one of them does not necessarily translate to success in another, and that's important. And we will come back to it in a minute. So what makes AI or an agent human-like, whether it's with regards to brains or behavior? Some people have suggested that it needs to pass some task benchmarks. They would define a set of tasks, for instance, Atari games, what have you, or set of numbers, handwritten, to be able to discriminate. And they say, okay, if you solve this task above some baseline threshold, then you're sufficiently human-like. But that's actually not necessarily true. Neither the behavior nor the mechanism is necessarily human-like in this case. You can solve the same task with architectures that are not brain-like at all. And the agent could show superhuman behavior that is not at all like humans, meaning it can make errors that are not human-like, or make no errors where humans would make errors. Also, behaving like humans on the same task is just not enough. For instance, there are the number of instances of speech that ChatGPT at this moment needs to hear equals 5,000 years of uninterrupted speech. That is not in any similar way the way that a human child learns and the speed at which a human child learns to speak. And the fact that there could be some similar behavior, again, does not mean that the behavior is fully comparable, nor does it mean that the mechanisms or even the training data were the same. Also, having partially brain-like mechanisms is not enough. For instance, convolutional neural networks that we have some experts in this room who have been working on it for a very long time. In the early 2010s, people suggested, okay, this explains the ventral visual cortex completely. However, when people started to look at things like adversarial noise, they noticed that there's something very different about these networks. If you showed the network this image, it would classify it as panda. And if you added this noise with very little amount, you would get this image that to us is really not distinguishable from the first one. All of a sudden, it would, with a large confidence, say that this is a given. On the other hand, there were adversarial patches. This is a banana, and this is a banana with a weird patch right next to it. Suddenly, this patch made the classifier say that this is a toaster. And so turns out that even though it seemed like it's a very promising, partially neurally plausible model, it wasn't behaviorally acting the way that we would expect something that is a model of human or primate visual brains to do that. Fortunately, there were some recent work that have been looking into more biologically inspired mechanisms, for instance, a particular kind of sampling that is a little bit more similar to how the retina might be doing it. And also more experiments with humans to see what are the cases where humans make errors in these kinds of adversarial attacks. Cases where it seems like, for instance, there is a point where you don't know if this is a cat or a dog, for instance. And those are interesting cases to compare. Very strangely, to date, there is no study that comprehensively compares the errors that humans make and the errors that vision models make with regards to adversarial examples in a comprehensive way. And that is yet to come, which shows that there's so much work to do, even though people have been working in this field for a long time. On the other hand, we have heard of AI that wins chess over humans, but some of my colleagues at Microsoft Research, including Sid Sen, with whom we are currently looking into using eye tracking to decode how many steps people are thinking, the depth of planning from their eye movements during playing chess. What they showed was that these superhuman AI chess playing agents, they actually do not generate human-like sequences. In the first study, they showed those sequences to experts, and the master chess players said this is not at all something that a human would produce. And in a second study, they actually, or in a third study, because already they had a couple of studies on this, they actually tried to improve the performance of human chess players by showing them the sequences that the AI had made. Turns out, it doesn't improve people who are average or higher, because those sequences are not helpful to humans in order to play their games better. So even when it shows some kind of a benchmark passing superhuman behavior, it actually doesn't guarantee that the sequences that it generates are helpful to humans or interpretable to humans. So let's come back here. I think this might have clarified these examples, what I meant by these different dimensions. For instance, superhuman chess is definitely serving the engineering benchmarks. It is not neurally plausible. It never had that goal. At the same time, I mean, I'm being not generous enough, but I haven't put it very high on the axis of being human-like in terms of behavior, because when people actually look at the behavior, they don't recognize it as human play and they can't learn from it. On the other hand, CNNs, for instance, they are very neurally plausible to a large extent. However, behaviorally, we see that they're not super plausible. Again, I'm not being very generous just to show the wide differences. I'm going to show you one example that is right over here, a human navigation Turing test next, to show you how, for instance, we have tried to compare the behavior. Another thing I want to say, note that these are non-binary evaluations. We're not saying it either is or it isn't. We're saying that there is some kind of a qualitative scale that one can imagine to just compare different studies. This one shows more human-like behavior than the other. This one is more neurally plausible than the other one. So here, I hope I can, I don't know how to play this other than bringing my mouse in, so I'm just going to do this. So this is the 4x4 game that we are using. And as you can imagine, 3D vision plays a role if anybody wants to play this. This is a game where spatial navigation is required, goal-directed navigation is required, collaboration and competition is required, and it was developed by Ninja Theory that joined Microsoft and Xbox in 2019. Why do we care here about this game? Because our goal is to create agents with individual and multi-agent human-like gameplay. So this is a 4x4 game. If you have only 6 people, can you play this game? If you have some AI that can come in and play with you. We have already published a couple of papers on this that you can see in ICML 2021, in CHI 2022 and 2023. And another one that I forgot to put there, iClaire 2023, where we try state-of-the-art deep learning agents or create new algorithms in order to test them on behavior on this game. And currently we're working on interpretable analysis of gameplay data. I'll tell you a little bit more about this. The game is 4x4, and there are 16 possible characters to choose from. So first we wanted to see, can we strip the game down, turn it into just navigation, and have agents that play like humans and test state-of-the-art agents and see whether they actually can play like humans. What happened here is we took the agents and put them on this map, stripped the game, and had one agent just walking around the map of the game. We used 2 state-of-the-art agent architectures. One was symbolic and one was hybrid. They were presented in a 2020 paper that was about deep reinforcement learning for navigation in AAA video games. What we did was we made sure, first of all, that these 2 agents were similar if you were comparing the kinds of metrics that usually are used. For instance, mean episode reward and time to, basically time to goal. And in both of those things, they were comparable. And then we asked what I'm going to ask you now, which is, which of these 2 agents do you think was controlled by a human, an average human, not someone who's a new player, not someone who's super advanced, and which one do you think was played by an AI agent? So I will ask, once they reach their goal, let's let our avatars reach their goals. Okay, so who thinks the video that is further from me is the one that is the correct one, that is played by a human? Okay. Who thinks this video that is closer to me was played by a human? I'm very glad we have so much variance. The video that is further from me was played by a human. That is correct. All right. I will show you 2 more videos. Between these 2, which one do you think was played by a human? Which avatar is controlled by a human? Let our agents reach their goals. All right, which one was controlled by a human? Who thinks it's the furthest from me that is human? Only 3 or 5 or 6, okay. Who thinks the one closer to me is human? Do you see how much more confident you are with this one? They were both agents. In fact, unbeknownst to our subjects, and subjects were sitting in front of the screen and watching these different videos, and they were supposed to tell us which one they think is more human-like. Unbeknownst to them, so 6 trials was really human versus agent, and 4 trials was the 2 agents versus each other. And this is the way that they responded. They chose the response, they said how certain they are, and here in the room, it was easier to see the certainty because in one case, in fact, in a case where there were no humans involved, you were sure. And as you saw, the 2 agents had very similar performance if you just looked at reward. Now let's see how our subjects saw it. We had 2 studies in our initial, this was replicated with much higher numbers, but I'm just gonna show you our first 2 studies. We had the first study, the human versus hybrid agent, were 6 trials, and hybrid versus symbolic, 4 trials. In the second study, people saw human versus symbolic agent on 6 trials, and on 4 trials was hybrid versus symbolic. So on the y-axis, you see agreement with ground truth. Ground truth is if they were always confusing. So they would pass Turing test if in the middle, at 50%, you saw most of the results being sort of gathered. And as you can see, it's much higher. In both cases, people are above 80% accurate in terms of detecting which agent is basically truly human. And when it comes to the hybrid versus symbolic, just like you, people were overwhelmingly confusing the hybrid agent with human. So when it came to comparing the two agents, just like the case that you did. And we tried to quantify the reasons for this. So in 2 other papers, we actually analyzed the explanations that people were providing. For instance, they were saying that the character is running smoothly, or the other one is more receptive, or the other one plays like me, which is why our current paper that we're going to present in 2020 in CHI is called Navigates Like Me. That was some of the things that people were actually offering as explanation. In a lot of my research, I actually study navigation, much more related to the previous talk. However, I'm not going to go into details there. I'm just going to say, on this rubric that we were talking about, HNTT, even though we did not design these agents to be neurally plausible, because we picked the state-of-the-art the state-of-the-art agents that were supposed to be very good at playing video games, and tested how human-like they are. And as you can see, in spite of the fact that we found these agents are not human-like, we are contributing to the dimension of human-like behavior. Because the goal of this was to show the importance of that in case somebody wants to play with these agents in this way. And then there is a term neuro AI, and I have summarized, and I'm going to have to skip faster because I was a little slow in the beginning. Building AI agents whose algorithms, representations, or functions are neurally plausible, or applying AI or behavioral models as hypothesis to test in neuroscience or as analysis tool for neural data, generating data sets or benchmarks for neural plausibility, or offering evidence to update or correct models in other dimensions can be summarized as neuro AI. That being said, I want to dissociate weak and strong neuro AI here as well. So weak neuro AI would be, okay, I just imported some model from another discipline and just tested it in neuroscience. I don't think that should be considered strong neuro AI, just importing something and testing, or just importing a model without changing it or without adding anything to it and just using it to analyze data. But it's still neuro AI, it's just weak neuro AI. The strong neuro AI would be a situation where there is true interaction and strong either bidirectional or three-way interactions across the different disciplines. And because we don't have time, I'm just going to mention the CNN example, the convolutional neural network example that I mentioned earlier. It covers this much of the space because it doesn't show human-like behavior. And in the paper, you can look at the sort of a very fast part of its evolution. Even though it started with inspiration from vertebrate visual nervous systems and led to a number of previous kind of iterations. And at some point, it was compared to neural data of IT vision. A lot of people here in this room are actually pioneers in this field. And then adversarial attacks arise, and it goes up to the sort of results that I was showing you that are more recent from Poggio Lab and others, that changing the way that the information is sampled can actually make it more biologically plausible and more robust to adversarial attacks. Because of these, at least the original CNN or convolutional neural networks have neural plausibility. They're useful in engineering, but they're not generating superhuman-like behavior. Just to go very quickly, I mentioned that there is interactions across these different ones. So a particular artificial agent, even with this kind of engineering goal, can be contributing to study of behavior or neurally plausible models. But it can also get inspirations from them, from those two other fields, or be corrected by them, or be tested by those methods. And all of them together sort of can create the sorts of work that some of us do, which is completely between them. I just want to say very quickly, what I'm not suggesting is that all engineers should drop what they're doing and do neuroscience, or that AGI has to use neuroscience, or that all neuroscientists need to work on AI. And to just go very quickly, I'm very out of time. Just very quickly, we tell you that we also tried, just to give you an example of the engineering goal, can we replace you guys by artificial judges? Can they judge just like you did, whether something is human-like or not? We gave different kinds of inputs, tried 6 different algorithms. Long story short, If I only showed them human versus agent, you could clap hands for me immediately and be like, great, you saved the day, you solved the problem, and I would be very happy. I'll show you the 0.85. However, if you look at the comparison of the two agents with each other, it's terrible. None of them can do this. So in other words, in spite of the fact that it might be able to do something on some benchmarks, you use a very similar benchmarks, you guys had no problems with it, it cannot produce similar behavior to what you just did or to what our participants did. So summary, state-of-the-art agents with the same benchmark don't show necessarily human behavior. Artificial judges cannot necessarily show human behavior. We have also done some work coming up in ICLAIR where we use diffusion models to imitate human behavior and doing some ongoing work inspired by task manifolds and prefrontal cortex on this topic. All right, summary. I propose a rubric to capture the main goals of research that are characterized as human-like AI, neuro AI, etc. And hope that these different dimensions can capture different ancestry and future of different projects. You can see which direction you want to go. And hopefully it's going to help people overcome some confusing crosstalk. With that, thank you so much.
I'd like to invite all three speakers to come and sit at the front. And we've got time for about 15 to 20 minutes worth of discussion. So if anyone has any questions, either in the audience or online, if you can, we've got people coming with microphones, so they'll. Oh yeah, if you could turn your microphones on. We'll see. We'll see which questions.
Let's just go straight.
Yeah. So if we can use the mics as well, because then people on Zoom can hear.
Do you want to use my mic?
Sure.
Okay, number one, presentation. I think that there was some framing effect, because the first question you asked, you gave only two choices. And when you asked the second, you don't think about the 3 or 4 options. Maybe there was some kind of framing effect because of the previous question that you asked. Number two, the first presentation, do you think what your research that we use cases of social justice and police brutality? Because there's a lot of bias among when police and minorities. So do you think we go there in terms of like preventing those kind of biases? Okay, thank you.
So if each of the respondents could also just quickly repeat the question.
So the question you were asking me was whether there was a framing effect between the questions that were being asked from the participants in the studies that I showed, the human participants. So every person received the same kind of question. It was not a different question. There was only one question, which is, there are 2 videos here. Which one is more likely to have been played by a human? That's it. There was no difference between the question in different cases. Unbeknownst to the people, sometimes the 2 videos were both played by agents, sometimes one of them was played by a human.
So they weren't given any choices, they just asked questions.
What do you mean by choices? There were 2 videos on every trial. There were 20 trials for you, for instance. You would come to the lab or in front of your computer, as this was Amazon Turk. You're in front of the screen. You see one video, you see another video, you choose one of them as being more human-like, you say why, and you say how confident you are. That's it.
Oh, me?
The question they ask you, the
Yeah.
second question.
So I'm not sure. So I'm not sure what do you mean by in my case. What was your question? I'm sorry. Again.
So in the current political sense, since this is science and society, one of the biggest issues in the United States is racial justice.
Yes.
So when cops see minority areas in Harlem or whatever, there's some kind of bias that they have. And there's a lot of police brutality, distrust. So can you research bias? You know, because the biggest issue is the freedom of vision that they have when they see an African American with a hoodie or with a clothing or something that's still something bad. So can you use it, you know, to reduce that kind of bias? Can it be applicable in society, in social justice? That's my question.
I mean, I wish I could tell you yes, that I could apply this research, but I think that for those kind of biases, probably we go beyond just a recognition of, you know, a 3D structure, for example, right? It's more of a, you go more of in the realm of pattern recognition, the fact that somebody is a hoodie, and then I would say more on the social psychology side of things. So, I mean, unfortunately, I don't think my research can give that much, and this is, I mean, but because it's very dry and it's all about how the brain just reconstructs 3D structures without meaning to these 3D structures. So.
Sure, wonderful, great, thanks so much. So I think we've got. Oh, have we got some more questions from the audience, and then?
The mic is not working, so you can take mine.
Yeah, we can share this one.
Yes.
Hello.
Did this work?
We'll
I'll
just
get
go
that
on.
back there.
Yeah, we'll go online until to the online questions until you get that there. Okay, here's a question for Kate. If you project the 3D grid cell blobs in the lattice onto the floor, i.e. only think about the XY position without Z, does it form a grid-like pattern?
Yeah, good question. And no, that doesn't.
The answer is no.
We did that. We projected it in all the different dimensions.
Okay.
Yep.
Thank you. Here's a question for Ida. How would one measure or quantify neural plausibility? The other two axes of your rubric seem quantitatively measurable, e.g. task performance, alignment with human behavior, variance explained in neural responses, but biological plausibility feels a little harder to measure.
I think some approaches that already exist are pretty great with that regard. One of them being some of the collection of available data from monkey electrophysiology and from humans, for instance, on object recognition. There is similar neural data available. For if you want to come to the domain of let's say spatial cognition or navigation, you have a huge amount of data from rodents, from bats, from humans, both neural and behavioral. So the very simple measure of that, the quantitative measure of that, is what extent of a battery of different sets of data sets within the field can a model explain. And I think there is, and I'm blanking on this, there is, MIT folks created this online tool with a lot of data. What is it called again? I keep forgetting. Brain something? Yeah. you don't remember either? What is that again? Brain score, yes, exactly. That's the term that I've been looking for. Fortunately, one of the world leaders in the field is here. Brain score, for instance, is one of those that has been created already. I think we need something like that for navigation, because a model that needs to explain navigation across different mammals, it would be reasonable to expect it to capture all of those different neural effects in the data.
Hi, this is a question for Professor Jeffery. About the cognitive maps, I was just wondering, so like in cases of like dementia and or Alzheimer's or any type of like neurodegenerative disease, what, if you know, because I know you're not studying humans, you're studying rats, but what kind of changes will we expect to see in those types of cognitive maps? Because we know that like sense of direction is almost always the first thing to go. And like, is it because there's less interaction with the environment, as you said, or is it just a matter of the neuron chemistry? And also between the rats, did you see any difference between like older rats and younger rats in their ability to create cognitive maps of their environment?
Yeah, really, really interesting question. So So yes, the discovery that the hippocampus, which is the structure that degenerates in Alzheimer's disease, so the discovery that that was involved in making this kind of mental map led to the question of, well, can we see navigation deficits in people who have dementia or pre-dementia? And there's a lot of work now to suggest that yes, people with Alzheimer's are very impaired in navigation. It's often the first symptom is that somebody gets lost coming back from somewhere very familiar that they've known all their lives, or eventually they can't find their way around their own house and so on. In normal aging, there's also some suggestion that navigation deficits might start to become more pronounced. Elderly people are less good at tracking their sort of position as they move through space. They're less good at what we call path integration and so on. And recently there's been some really interesting work, some of it here by Josh Jacobs and his colleagues, so it's been found that we can sort of see a signature of grid cell activity in human brain scans. And it's kind of technical how they do this, but when those brain scanning methods are applied to people who are at genetic risk of Alzheimer's disease, abnormalities in that signature, that grid signature, are seen, suggesting that long before symptoms develop, there may be problems developing in this navigation system and this sort of representational system. So I think that's super, super cool work. So yes, there's really strong links between the navigation and dementia.
Good. So if there are any questions in the audience, if you raise your hands, don't be shy, put your hands up high. I think we've got a couple over here, and then we've also got some more online.
Thank you for your presentation. So this is a question for Professor Jeffery and Professor Domini. So I am a PhD student working on robotics. After listening to Professor Jeffery's presentation, I have a question, because in your experiments, the rat is actually navigating in a maze. So it's a quite a limited environment. Do you imagine we can extend this kind of like a cell structure to a worldwide scenario? For example, if the rat is actually navigating on the Earth, it's like a very bigger maze. In that case, will this kind of structure gonna be the same, or do you think that this kind of structure will be reused when the rat navigating to another area? And it also reminds me of some of the current research in the robotics, because currently people are thinking how we can have inherent word models for the robot to navigate in the world. So that relates to Professor Domini's presentation. So my question is that, should we try to mimic this biological structure while we are designing these computer vision or robotics navigation algorithm? And how do you envision that we can do that? Thank you.
Shall I take the first part and you take the second part? So the question of how big is this map? That's a really interesting question. So does a whale, for example, have huge place fields out in the ocean and all of this kind of stuff? So, we record from rats and mice in very bounded environments with walls, and we find that the cells are very kind of sensitive to the locations of the walls, and they use the walls to help position the places where they fire and so on. But what happens in the real world if you're in a field or something like that and you haven't got walls? And we don't know very much about that yet. We do know that that in larger areas, place cells will just produce more fields. So in a small space, they may just have one or two. In a larger space, they will tend to have more. There's some really beautiful work coming from Nakhon Monosky's lab in Israel, where they've been studying bats, which fly really long distances to go foraging at night, and they're starting to study what happens to the place in the grid cells over these really long distances. And I think the work's not complete yet, but they're seeing evidence that you can have really huge place fields. So it looks like there's multiple scales of these representations. So I think probably what the brain does is it creates a sort of hierarchical set of different scale maps for different situations. So I've got one map for this room, I've got a bigger map for the building, I've got a bigger map for the city, and so on and so on. so I'm using sort of a different type of representation depending on the scale. But we don't know very much about how the brain is representing the places that you're not in right now. So there's a whole set of questions there we haven't answered yet.
Is there another question back there?
Okay.
Sorry, Phil.
Yeah,
excuse
yeah.
me, go ahead. Yeah.
Can you hear me? Or?
Yes.
Okay, so I mean, the answer is yes. You should give it a shot and try to emulate more biological systems as engineers. And the reason why I'm saying that is not because that is leading necessarily to better success. In fact, you build airplanes without really mimicking how birds fly, right? And that works fine. But I think that maybe by being inspired by biological systems and rejecting some assumptions about what I said before, the fact that the brain reconstructs the three-dimensional structure of the world as the physical structure, this kind of copy in your brain, right? This comes from computer science. So we as psychologists took these models and said, oh, let's see if the brain does that. I mean, I think it doesn't at all. And maybe going back to computer science, there you could say, well, we can maybe reject this as a goal of the system and try to see which other goals can be achieved and how you can achieve goals that are mimicking humans without having to calculate certain things you calculated in the past. I don't know if the answer is clear enough, but that's I think being inspired by biological systems can actually change the way you look at things in computer science. So can be inspiring in that way. So
Can I add something to that?
Very quickly.
Very quick, yeah. I recommend taking a look at RATSLAM, which is, and also successor representations have been used by roboticists at Stanford in order to do also navigation. And that's something that my work has worked on, which humans and rodents seem to use for in the hippocampus for navigation, and their eigenvectors actually capture what the entorhinal cortex is doing in grid fields. So take a look at those.
Cool. So I think we've got time just for two very quick questions. So one person was trying to ask before, so we'll let them ask their question, then we'll take one from online. Thanks.
Thank you. This question is primarily for Dr. Momennejad, but maybe for the whole panel, about the effects that the encoding of 3D space has on the behavior of AI agents. So specifically, I was wondering if the AI bots that you discussed have to reduce the 3D structure of the environment to 2 dimensions and then reconstruct the 3rd dimension, or if they operate in an explicitly 3D environment.
Thank you for asking that. So both of the agents that I showed you, they do use 3D information. However, one of them uses symbolic information, which just is like X, Y, Z, etc. The other one used depth of field as well as the entire voxel space. So, and there is some pre-processing that happens when it's looking at the kind of very rich number of, huge number of pixels that's in front of it, that it actually does, for instance, estimate depth. So depth is actually one of the main features of the agent that all of you thought was more human-like, which kind of relates to your work a little bit. So it's very important. And it turns out just symbolic information about where things are were not enough. And it was important to have that vision component where the agents could infer certain things about the depth and about various other features from the pixels in the vision, as opposed to just receiving certain kind of things about X, Y, Z and where they were and some symbolic information.
Cool, thanks.
Great. So we have an interesting question online, which we'll ask very quickly. Could the speakers comment on any common theme that there is between their talks? Paul emphasized the idea that 3D vision might not be based on a fixed internal representation. For example, the observer's apparent representation varies with the task. But Fulvio's proposal does not seem to be task-dependent. Kate's talk emphasizes metric representation rather than task dependence. And I'm not sure how task-dependent Ida's representation is. Is there a different common theme? Take it away. Yeah.
Thinking.
So I have a lot of work that is a lot more related to Kate's work, basically, but in the humans, in the human domain. I don't think it's metric. because of the fact that, for instance, place fields, etc., gets sort of somehow more pronounced around areas of goals, and even grid fields change over time. So there are some reasons to believe that, and models that we have tested that capture behavior are also like that. In our agents, the depth and the 3D actual vision did matter. It did make the agents, even though none of them passed the Turing test, at least in comparison, they looked more human-like, and depth was something that Fulvio was talking about. In terms of the representations, it is possible to show behavior without having the internal representations that are neurally plausible. However, when you compare across tasks, as I was showing in one of the agents, it doesn't necessarily generalize to another task. And I think having those neurally plausible representations are actually required if you want to have a battery of different tasks that the agent needs to address.
It was a question of common themes, whether task dependence might be a common theme or whether there are other common themes.
I mean, yeah, actually, I was pleased that there was a lot of common themes here. I guess that I would say that in general, we have certain preconceptions about how the brain should work because we have learned how the world is as scientists, as humans, through evolution and through culture, right? And so we think that the brain does the same thing. I think that's my take of this. And in fact, that's not what is going on. So the brain can be very different from us. That's my take.
Kate, do you want to comment?
Yeah, I think the common theme is that these representations aren't metric. I agree, but I also would say that they're not metric either. There's clearly, we've got head direction cells, we've got grid cells. They're clearly doing some processing of distance and direction. It's just not rigid always. Although the head direction cells actually are pretty rigid, but the grid cells less so. So it really matters which way you're facing, but not so much about how far you've gone. But yeah, also I think the fact that the representation or map or whatever you want to call it, is constructed according to the limitations of the environment. I don't know how much I'd call it task demands as opposed to environmental constraints. Maybe it's both. I don't think we know. But certainly, I think we're letting go, if it was ever really a thing, of the idea that that the brain is trying to create an accurate map of what's out there, because that's not the function of the brain. The brain is not trying to represent the outside world. The brain is trying to make you do things that will enhance your survival, and that may involve sort of representing what's out there in the world. But if it's not important for it to be accurate, the brain doesn't make it accurate. The brain is doing what it needs to do to make you behave adaptively. That's the ultimate message, I think.
Fascinating. I wish we could take all the questions and continue this conversation that got us into some really interesting territory. Would you like to have the last word?
Well, look, thanks so much for everyone for turning up. It's been a fascinating discussion with the three of you. We've learned a great deal about human vision, animal navigation, and AI. And I just want to say, do sort of look at the volume and enjoy it, but also join us for a reception now to further discuss, and do find the speakers and ask them some pressing questions. Thanks a lot.